Our second speaker, who's being wired, is Dr. Larry Lasky. Larry is our Renaissance man. Larry plays the timpani and percussion in 2 symphony orchestras when not scuba diving or traveling the world with his family. In his spare time, Larry is also the venture partner at U.S. Venture Partners out on Sand Hill Road in Menlo Park. As some of you may recall, Larry is also one of the great leading lights of the biotech industry. From its infancy, starting off with the Genetics Institute in the early 80s, and then with Genentech for nearly 20 years. Larry is going to talk with us today about different approaches taken by early-stage biotech companies that are exploring a variety of oncogenetic pathways involved in involved in large numbers of tumors, presumably simultaneously, or a magic bullet of sorts. Is that? We'll see.
Well, I was going to stop it at Renaissance Man, but let's...
Okay, should we go back to the Renaissance Man?
Stop it at Magic Bullet.
We will start with a solo percussionist here.
Thank you for the overly kind introduction. First, I'd like to thank my old buddy Larry Gold for inviting me. I've known Larry a long time, and I want to assure him that shots, cheap or otherwise, will not be made during this talk. I know that's a huge mistake to do with him, so that will not happen. Now, at dinner last night, somebody said, I looked at your slides, and they look pretty complicated. So clearly, I got the message from Larry too late about simplifying things. I was going to run out and buy 100 copies of Molecular and Cellular Biology for Dummies, but your Boulder bookstore was out of them. So, but I want to, assure you that you have to have a sort of focused, open mind here. I want to assume that the reason the slides seemed complicated last night was because he didn't have the benefit of me helping him through them. But we'll see how the experiment goes, okay? Now, we all know that oncology and cancer are still huge unmet needs. It's still the biggest killer in the Western world by far. That is in large part because many of the medicines we still use are what I call old school medicines. And essentially what physicians started out thinking was that, well, cancer cells grow fast, so if you kill them faster than you kill the patient, it'll have benefit. So they produce cytotoxic drugs, and cytotoxic drugs are basically poisons which have a modest therapeutic index. But they have problems. Many of them we have unclear mechanisms of action. In other words, we don't know what the targets are. That's a major problem. A second problem is that they have poor therapeutic indices. So while they do kill tumor cells, they also damage normal cells, especially rapidly dividing cells such as gut and hematopoietic cells, blood cells. A real problem is the rapid emergence of resistant tumors. So tumors very quickly become resistant to the forms of chemotherapy that to these cytotoxic chemotherapeutic drugs, but we don't understand why. And of course, personalized medicine, genomic analysis, all these analyses, omics that we're hearing about, will in the future help there, but they don't really help now. So all this sort of ends up being a problem. The initial response rates are relatively high, but they fall rapidly and they're very unsatisfactory. Now, what this has led to is the idea of pathway-specific tumor therapies. And people diss basic research. Oh, why do you spend a lot of money on flies and worms? But it turns out that all of these pathways were initially discovered in simple genetic systems. Drosophila, the fruit fly, C. elegans, the nematode. All of these pathways were discovered there. So once they were understood in these pathways, once they were understood in these lower organisms, we could then show that they also were found in higher organisms, rats, mice, and us. Now, what tumors do, as tumors like to do, they co-opt these pathways for unregulated purposes, growth and survival. And the way they do this is by mutating the pathways. So they either mutate the DNA or they change the landscape that the DNA exists in. They have many different ways to do this. So that suggests that the tumors are doing this for their own selfish purposes. Now, Tom talked yesterday about the first example of a pathway-specific medicine, which was Herceptin, produced by my old company, Genentech, now Roche. I hate saying that. Excuse me, the willies. I'm not there anymore, so. A second great example is, and Larry and I were talking about this last night, the so-called miracle drug Gleevec, which is used in chronic myelogenous leukemia, which is an absolutely astonishing drug, which acts on a translocation where two proteins come together and are involved with allowing the tumor cell to exist and to proliferate. Now, what I'm going to talk about today is the hardcore science behind 3 companies which are trying to develop more of these types of medicines. Now, the way we're going to do this is we're going to talk about the pathway first, and then we're going to talk about what the company is doing in the pathway. Now, don't be worried about these pathways, because actually they're just blobs of proteins that interact with each other and do certain things in cells. The first one we're going to talk about, the first company, is Intellikine. And the first pathway is the famous Pi3 Kinase Pathway. Now, this is pretty much what all these pathways look like. See, it is simple. Basically, here's the outside of the cell, and here's the inside of the cell, and these blobs are various proteins which interact with each other and do certain things. In this case, what they do is they produce normal growth and survival signals for the cell. This pathway is involved with many different proteins, as you can see, from proteins that are on the surface of the cell to proteins that are inside the cell. What's fascinating about all of these proteins is that they're highly mutated in many types of tumors. So this is a survey, a current survey of the mutations of these proteins in tumors. These mutations have been determined from primary tumors, from tumors from humans. So they're actually mutations that are involved with some advantage that the tumor has. Now, you can see in some cases, as much as in colon, for example, one of these proteins called AKT, 46% of the tumors have mutations. There's other such examples. In the case of endometrial cancers, another protein in the pathway called PTEN has 50% mutations. In another case, sarcomas, almost 80%. So you can see these tumors have these mutations for their own purposes. Now, what Intellikine intends to do is target these various components which are mutated in these cells. Now, why would they want to do that? Well, the assumption is this pathway is involved with the inappropriate growth and survival of tumors. They are mutated in tumor cells. Therefore, if you inhibit one or more of these proteins, you will have a beneficial effect on tumor treatment. And they've picked several of these proteins. Pi3 kinases, which are the proteins that are proximal to the surface of the cell, and the one I'll talk about today called TORC, which is target of rapamycin, which is an extremely interesting protein. Now, it should be said right now that all of these proteins do similar things, which is they take a phosphate, the atom phosphate, and they add it to various other proteins, which then regulates the activity of those proteins. They are all, therefore, kinases. Now, the important thing, skip that, the important thing about this philosophy of drug development is really highlighted on this slide. When you pick a pathway and a target within that pathway, you can develop an extremely high degree of specificity for that target. And if you assume, and we'll talk about this in the second vignette, I'm going to talk about proteolytics. If you assume that target specificity translates to higher efficacy and higher safety, then this is a very, very clever way of making new cancer drugs. Now, illustrated on this slide are so-called dendrograms, which are trees of proteins. And on these trees are 400 different proteins in the family of the target. So I told you these are kinases, proteins that add phosphate. These are 400 different kinases, okay? Now, these are all approved drugs. Where you see a red ball, that means the drug hits another protein. The most specific is Gleevec. You can see it here. It only hits the right target, but it also hits other targets. And the closer to the ball it is, the more closely related the kinases are. So it hits kinases that are closely related to the target, but very little else. This thing hits like tons of things, yet it still has some efficacy. Now, there's safety issues with these kinds of drugs. Okay? Now, if you'll notice what Intellikine has done, they have developed a tremendous level of specificity. And that's what you can do. It only hits the appropriate target here and another target modestly far away. Now, with this kind of science, you can therefore develop very specific drugs for the targets of interest. When you combine that capability with the fact that these targets have been validated or semi-validated by genetic systems as well as by mutations in humans, you have a very powerful drug approach. And that, in a nutshell, is what we're going to talk about today. Now, this is probably one of those slides that freak the guy out, but it's actually quite simple, okay? And these, you'll see many of these. All right. Now, what this is basically, this is time. We all know what that is. And this is the size of the tumor. Okay. So what you can see here very clearly is this drug does something very interesting to tumors. It stops them from growing. This is in an animal model. So if you take these cells and put them in mice, they grow very rapidly, but in the presence of this drug, they don't grow at all. Now, this is the fancy name for this experiment is a xenograft. My name for this experiment is a hairy test tube experiment, because that's basically what it is. It suggests that it might work in humans, if you assume these cells are remotely like the kind of cells that you'll be hitting in humans, but in fact, it's just an experiment. But it's nice that at these drug doses, you get an inhibition of tumor growth. What these blobs show is the ability to dissect the components of the pathway and the effect of the drug on the components of the pathway. And that's very important, because dissections such as this can be done in human samples. So you can take biopsies out of patients before and after treatment and do such dissections. When there's a big blob, that means the tumor's happy. When there's no blob, that means the drug is working. And you can do these kinds of surrogate studies in humans. So this is very important. This is very different than dumping a cytotoxic into a person and seeing if a tumor shrinks. Okay, let's go to this. Now, second to that is experiments which show that with actual tumor cells in animal models, you can show an effect of the drug. And that's what's shown here. Now, this is this is quite simple, okay? These are the sizes of spleens of mice that have been given human tumor cells, acute lymphocytic leukemia cells, okay? These are primary cells, straight out of a patient. And what you can see is their spleens become enormous. Now, when you treat with Gleevec alone, you see somewhat of a decrease. The spleens are getting smaller. But when you treat with Gleevec plus the drug Intellikine's trying to produce, the spleens become normalized, healthy spleen weight. This is a better experiment than the one I showed previously because they are using primary cells. So it's a much more compelling experiment. All of this has led to Intellikine putting this drug into a phase 1 trial. So this drug for mTOR, for that target, is currently in a phase 1 trial, which is for safety, we hope for efficacy. Now, we'll get into this a little more at the end. But of course, if you know, you remember the slide where I showed certain classes of tumors are highly mutated in these pathways. Obviously, what we're trying to do is get patients from those classes of tumors into a phase 1 trial who have those mutations in order to try and see efficacy. That's another very powerful aspect of target-specific medicine. You can pre-select patients who have these mutations and put them into your clinical trial. Okay. Now, it's great to shrink a tumor in a mouse, especially if you're a mouse. But big pharma would, I know there's big pharmas here and they're going, okay, who cares? So now we're going to talk about real clinical data. And this is a very exciting story. It's the story of proteolytics and the drug carfilzomib. Now, this is another pathway. This pathway, which has generated a few Nobel Prizes, is a fascinating pathway. In the old days, people just thought, oh, proteins are made and they're degraded in this kind of unregulated way, and there's this turnover and ho-hum. Well, it turns out it's far more complicated than that. And it's a highly regulated pathway whereby a protein called ubiquitin is added to the protein to be degraded. The so-called marked protein is then recognized by this giant structure, which essentially looks like a garbage can, and it is a garbage disposal. It's the proteosome. And the proteosome is a very complex structure that degrades proteins that are marked. And this is a very highly regulated pathway. Now, when this first came out, I thought you will never be able to make a drug against this pathway because this is far too important of a component to the cell. If you try and inhibit this, it'll be too toxic. Well, Julian Adams and Millennium didn't believe that, and they actually produced a drug called Velcade, which is about a $1.3 billion drug, which is fantastically effective in multiple myeloma. Now, why would one go up against a drug that's already effective? Well, it turns out that Velcade has a profound problem, and that problem is a side effect issue. The side effect issue is that a large percentage of the patients who get Velcade get peripheral neuropathy. Peripheral neuropathy is a painful side effect. It's so painful you can't walk, you can't touch anything, and it's dose limiting. Many patients will go off the drug. Now, why does that drug have that side effect? Well, there's two possibilities. One is that it's an on-target side effect. That is, when you hit this proteasome with a drug, you cause peripheral neuropathy. That's one choice. A second choice is it's an off-target side effect. Now, you remember the dendrogram I showed where you can get very specific inhibitors of pathways. Well, let's see how specific Velcade is compared to the drug we're going to talk about. This is carfilzomib. Now, carfilzomib is a compound that came out of a lab at Yale, Craig Crews, which was shown to be a proteasome inhibitor. So just like Velcade, it inhibits that garbage disposal exactly the same way. It looks very different from Velcade, though, structurally. Now, what was very exciting initially about the data from Crews, from that lab, was the following experiment, which is what sold me on starting the company. And this, again, is like a dendrogram, but for proteases. That is, for proteins that degrade other proteins. And what you see here are a collection of proteases. These are all different proteins that degrade other proteins, broken up into classes: serine proteases, cysteine proteases, metalloproteases, et cetera. Here is the proteasome itself. Now, and in blue is our drug, and in gray is the competition. Now, not a surprise that both very effectively inhibit the proteasome. But what is a surprise is when you start to look at other proteins. What you can see is that the competition is hitting lots of other proteases, and profoundly in some cases at this concentration. Ours is much more specific. Okay, you can see very little activity, maybe a little bit in chymotrypsin, but very little activity. That's what sold me on this. I assumed that specificity would translate to decreased neuropathy. That was the idea. It costs $140 million to prove that, but that's how we roll. Now, so here's the preclinical profile of carfilzomib. Had minimal off-target activity, consistent with better side effect profile. It had an irreversible mechanism. So it bound to the proteasome irreversibly. It stuck there and didn't come out. So that's another possible advantage in that it would stay there and inhibit the target for longer. And so those two components were major drivers towards putting this in the clinic. Now, the drug has been in hundreds of people now, and I'm going to show you some of the early data. So these aren't mice. These are real people with real myeloma. Now, this was probably one of the, so I've been doing biotech for 30 years. I'm older than Larry, it turns out, actually. No, I'm kidding. Is anybody? No, so, uh-oh. Oh, man. Oh. Whew. That was stupid. All right, so, all right, so, we go way back. All right, so this is a patient population which is very, very sick. This is the relapsed refractory myeloma population. And this is a population who have seen all of the drugs, Rev, Dex, thalidomide, bortezomib, all of them. So they really don't have any other choice. Now, what you see here are the various subdivisions that we put patients in when they're treated with the drug. And what's most exciting is this. This is minimal response and partial response. That means that in this very small trial, about 26% of the patients with no other recourse responded to this drug. This was very exciting. This means that this has a chance of being a drug. This was the first data to suggest that this could be a drug. Not only did they respond, but the responses are durable. These, remember, are very sick patients. The median time to progression was about 5 months. So not only do they respond, but they respond for long periods of time. Okay, these are patients that have no other drugs and who will be dead long before this. Okay. Now, this is probably the most interesting part of the experiment, which is the neuropathy rate. Now, again, these are small numbers. Obviously, the numbers are much larger now. We've treated hundreds of patients. But the data have carried through. The neuropathy rate for Velcade can be as high as 30%. What's striking here is this is grade 1, grade 2, grade 3 neuropathy, extremely low. And this has carried through. The neuropathy rates are 1% or less. So, and a lot of this is luck, the concept that higher specificity would result in better side effect profile seems to be the case, which is very important. This allows this drug to compete with Velcade. Okay, now a little more data, and then we'll get to the last. I can't believe I'm on time. All right, so what about if you treat patients who are less heavily treated? So this is the so-called relapsed population, and these are patients, some of whom haven't seen Velcade. Now you can imagine it's very hard to treat a patient that's already seen Velcade and who has progressed on Velcade. It's hard to imagine how they would now be sensitive to our drug, which hits the same target. In spite of that fact, some of them are. But what about patients who have never seen Velcade? And this is a trial which has both of those types of patients. Bortezomib is the other name for Velcade, bortezomib naive and bortezomib exposed. And what you can see here, and again, these are small numbers but are much bigger now. Almost 60% of these patients respond to our drug. This is single agent activity. That's important. We were talking before, somebody yesterday was mentioning, oh, everything's going to have to be used in combination. This is single agent activity. No other drugs, just this. The combined overall response rate was close to 36%. That included bortezomib exposed patients, Velcade exposed patients. So again, this looks very good. An overall response rate in a heavily treated population of almost 36%. Again, the time to progression was also impressive. In this case, these are less heavily treated patients, so I think it was 9 or so months time to progression. These are really exciting data for people with myeloma, late stage myeloma. Again, here are the neuropathy rates, and you'll see they're strikingly low. Grade 1 and 2 basically are almost asymptomatic. Grade 3 is about 3%, but these are very small numbers. This is one person. It could be 0, it could be 2, right? These numbers are holding up, though. So here are the conclusions. Single agent carfilzomib is highly active, very well tolerated. In fact, we are continuing our dose escalation. We thought we had our MTD at 27 milligrams per meter squared. We're going higher. This is almost like, I don't want to jinx it, but it's kind of like PBS. The patients don't believe they're getting a drug, which is unusual, but it's a very specific drug. The responses are quite durable. The AEs are manageable. And peripheral neuropathy just does not seem to be a problem with this drug. So we have eliminated, we think, and we have to do more patients, more trials, we think we have eliminated a major side effect of treating myeloma with proteasome inhibitors. Okay, well, now we're going to go to something way out there, which is Oncomed. Now, I teach a class at Berkeley on how to start a biotech company. I bin things into low risk, medium risk, and high risk. Proteolix was low risk. IntelliKinds, medium risk. This thing is like way out there for risk. But I think you'll agree when you hear the concept, it's, if true, one of the more exciting new areas of oncology. Now, we've known for a while that when we treat tumors, they come back, no matter what we treat them with. And when they come back with a vengeance. And they are resistant when they come back. And there's two possible hypotheses there. One is that every cell in the tumor is capable of forming the tumor. The second is the hypothesis, which was originally proposed by John Dick in hematopoietic cells and then carried through by Michael Clark in solid tumors, that there's a subset of tumor-initiating cells or cancer stem cells. I don't care what you call it. They're formally the same thing, either a cell that can initiate the tumor that's different from the rest of the tumor, or a stem cell that can do it. Formally, they overlap. Now, if you look at a tumor with a marker which subsets cells which contain tumor-initiating cells, this is a pancreatic tumor from humans. What you'll see is there's a lot of heterogeneity. Some of the cells have it, some of the cells don't. And so there's heterogeneity, and this was identified early on. What Clark did was to take advantage of this heterogeneity and fractionate tumors using traditional FACS, activated cell sorter technology, into various fractions with these markers and test their tumorigenicity in animal models. And what he found was that there was a small subset of cells which caused tumors. And this, in essence, is the cancer stem cell or tumor-initiating cell hypothesis. The tumor is a heterogeneous collection of cells, most of which are differentiated cells, which are incapable of forming a tumor, a subset of which are stem cells, which are the bad guys, which form tumors and which seed metastases. That's the hypothesis. Now, what we've been doing all along is getting rid of the differentiated cells. So here you see we treat them with cytotoxics, we treat them with all kinds of things, which shrinks the tumor, but lo and behold, the tumor always comes back. That actually, and we'll have data to support that, is consistent with what we know about stem cells, which are remarkably resistant to toxins. And of course, evolutionarily speaking, you need your stem cells to be resistant to toxins because you don't want them to go away, or else you go away. Your skin goes away, your gut, or your blood goes away. So that seems to be the case with tumor stem cells, and I'll show you some data to support that. Now, what Oncomed is trying to do is kill or eliminate these cells. Once these cells go, the tumor will just degenerate. Or if you treat with chemotherapy, it won't come back. So that's the idea. Extremely interesting and difficult and controversial. Now, what's interesting about, wow, the cells got left out. Well, there's supposed to be cells here. What's interesting about these tumor stem cells is that they have the same pathways which normal stem cells have, only they're deregulated. So we know from Drosophila, from C. elegans, from mice, from lots of low organisms, that there are many interesting pathways which are involved with stem cell maintenance and controlling differentiation of cells. And it turns out that these cancer stem cells utilize many of these same pathways, although for their nefarious means. The pathways I'll talk about today are the Notch pathway, which was named after a mutation in Drosophila, and the Wint pathway, which was named after an integration site mutant that Harold Varmus found 25 years ago. So these were done in other species, not in humans, yet they now have come to fruition in humans. Now, here is one of the pathways, and this is the problem with these developmental pathways. And this is why this is probably the most difficult thing anybody's trying to do in drug development right now. They're ferociously complicated. They're not just one protein binding with another protein, like EGF, epidermal growth factor binding to epidermal growth factor receptor. There are many components to these pathways. That's highlighted here in the Notch pathway, where there are 5 ligands, like epidermal growth factor, and 4 receptors. And these all work in combinatorial ways. And some are expressed on the same cell, some are expressed on different cells. And these components regulate differentiation in animals. But there seems to be some deregulation of these in these systems in tumor cells. Now, the first thing that Oncomed focused on was a protein called DLL4 for delta-like ligand 4. Again, that name came from a Drosophila mutant. So we're going way back to old days for all these discoveries. And what Oncomed does, and they're probably the best in the world, Genentech / Roche notwithstanding, is they make monoclonal antibodies like nobody's business. They are unbelievably good at making monoclonal antibodies which block the various activities of these proteins. They are absolutely amazing. So they have made a monoclonal antibody against DLL4. Their assays, their xenograft assays actually are different from the ones I talked about earlier. Theirs are less hairy test tube assays because they use primary human tumor cells and they subfractionate the so-called tumor-initiating cell subset to do their assays. So the assays are far more realistic, although there is still controversy about the assays. It's always hard to put human cells into animals. Their assays are far more realistic. This is a classic Oncomed experiment, which shows that a colon tumor grows rapidly in mice, although you'll note it grows slower. And the reason it grows slower is because it's human cells. It's not a cell line, so it grows much slower. This is 5 times slower than the previous. Here's the tumor volume. And what you can see is that the current standard of care for colon cancer, irinotecan, does a pretty good job of knocking it down. But when you combine irinotecan with the antibody against DLL4, it just wipes out the tumor. The tumor's gone. And we'll have more data to support that. It essentially blocks tumor formation. Now, this is the so-called Oncomed bounce back experiment, which me and a few other guys came up with at the beginning. And it's a fascinating experiment. What you do is you let a tumor grow, and then you add the antibody of interest, DLL4 or control, plus a drug. And those shrink the tumor. You then remove the drug, but leave each antibody on the animal. Now, if you're really killing the tumor stem cells or tumor-initiating cells, the tumor shouldn't grow back. And that's exactly what happens. You can see in the presence of the antibody alone, the tumor is wiped out, whereas with the control antibody, it just grows right back because you haven't killed the tumor stem cells. So this has become a classic experiment now to show that your antibody is in fact having a profound effect on these tumor-initiating cells. Now, Oncomed, they're a little crazy there. They do stuff that not even I would do in my right mind. And one thing they do is actually measure the ratio of tumor stem cells in the tumor. So they take out the tumor and measure using dilution how many cells are there. And that's shown here. So in the control, untreated animal, there's about 1 tumor stem cell for every 173 cells. Okay, that's the ratio. When you treat with their antibody, that ratio drops profoundly to 1 in 441. Okay? That suggests that you're killing 70% or more of the tumor stem cells. Here's an interesting experiment. When you add the irinotecan, the ratio goes up. That's the key. Larry got this. You're shrinking the tumor, but you're not killing the tumor stem cells. And this has actually been observed in many situations, glioblastoma, et cetera. The tumor stem cells seem to be resistant to chemotherapy. Importantly, when you combine irinotecan plus the antibody, now your ratio is down to 1 in 719. So this is consistent with the data that we saw earlier. So this antibody is now in the clinic. The difficulty with putting these kinds of things into the clinic is it's never been tried before. You don't know anything. You can do all the talks, you can do all the preclinical efficacy you want. You don't know what's going to happen. There's been a lot of ups and downs, but we're now learning how to use this antibody. It's very interesting. It's a single agent study. There's some hints of things going on, but way too early to tell. And it's in phase 1 right now. This whole program has been partnered with GlaxoSmithKline, I should say. Now to finish up, we'll talk about something that's very interesting with respect to another pathway, and this is the Wint pathway. Now the Wint pathway has scared the bejesus out of a lot of people because you think Notch delta is complicated. This is way worse. The receptors for Wnt are the frizzleds, again, Drosophila. These are proteins that go through the membrane a bunch of times. There's about 8 of those, maybe more. The ligands are the Wints, and there's, I think, 15 of those, something like that, depending on the organism. And again, these act all combinatorially. And, you know, it's crazy what we're doing, but it's exciting. It looks like something's going on. And you don't need to know what goes. These are just very complicated things that go on intracellularly. Basically, what we're trying to do is block the interaction between this and this. That's the idea. Now, Oncomed has, we know for sure, made the first in class Wnt pathway antagonists. They have both monoclonal antibodies that hit various frizzled components, various receptor components, and they have Fc receptor chimeras. These are another kind of inhibitor which soak up the ligand. So they have the first ones. We know it because Genentech's desperate to get a hold of what we have, and if anybody was going to have it, they would. So this is very exciting, but very complicated. Now I'll finish up by showing, again, the same kind of data with one interesting twist. Here you can see this is a breast tumor with standard of care here. So again, tumor volume days. Standard of care for this breast cancer, it's an ER positive, is Taxol. And you can see there's some effect of Taxol. And interestingly, with the Wnt antibody alone, you see very little effect on tumor growth. However, if you combine these two agents, you see a profound inhibition of tumor growth in the mice. So this suggests that, as with DLL4, Wnt, this particular frizzle, this is against the receptor, this particular frizzle is somehow involved with tumor growth. Now, this is probably one of the most amazing results I've ever seen. Okay, I've seen a lot of results. So this is pretty amazing. Okay, now this is a pancreatic tumor, which has been treated with that same antibody. Okay, now follow me here. Here is the control tumor, and what you can see here is there's all the ducts. So here you can see the ducts. And here is the control tumor treated with the crappy standard of care gemcitabine, which is one of the worst, most poorly efficacious drugs out there. And doesn't look that different. Okay, now when you add Wnt antibody to these animals, you see a profound change. The frickin' thing turns blue. Now, why? It's not blue in the animal. We've stained it blue. And what we're staining here are mucin-containing cells. Now, it's known that mucin-containing cells line these ducts. You can see them here. But when we treat with the antibody, we essentially force the tumor to differentiate into a big hunk of mucin-containing cells. And you can see with gemcitabine, you see the same thing. Okay, so this is this idea of tumor differentiation therapy. It's been done for a long time with retinoic acid responsive tumors, but hematopoietic tumors. But this is one of the first cases where it actually might be useful in solid tumors. Now, you know, one could say, well, yeah, but it's still a big tumor. Are there any tumor stem cells in there? Well, fortunately, there's not. So again, you see this same experiment where you look at the frequency of cancer stem cells. Here are the control antibodies. They're about 1 out of 106. Here is the anti-Wnt, anti-frizzled antibody, about 1 out of 319. Again, gemcitabine kills normal cells but doesn't touch the tumor stem cells. And the combination wipes out the tumor stem cells. I think it's 1 in 1,200. So even though this thing is a big hunk of mucin, it's differentiated. This is hard to get your head around because we don't know what's going to happen in people when we try this. But nevertheless, that's that's the data. Seen enough of those. Okay, so here's the summary. Several developmental pathways originally elucidated in worms and flies and other models are important for self-renewal, this idea that stem cells like to replicate themselves without differentiating and throw off another cell which can differentiate. That's the definition of self-renewal. Oncomed probably has the most diversity of interesting reagents to look at these developmental pathways. They're working on other pathways which are much earlier. They've shown marked activity in novel animal models using primary early passage human tumors. That's a very important distinction. Of course, nothing is as good as the clinic, but this isn't bad. DLL4 is in phase 1, and Wint is being prepared for phase 1 trials for next year. All right, so let's summarize the talk. This is a personalized medicine kind of meeting, so I want to loop back around there. So, these genetic, that is DNA mutants, as well as epigenetic, that is the sort of milieu, the environment that the DNA lives in, changes in tumor cells are involved with the dysregulation of many developmental growth and survival pathways. I've just talked about 3 of them, there's many others. This allows for various new technologies. All these omics, epigenomics, transcriptomics, proteomics, et cetera, can allow you to subset patients into classes which have a higher probability of responding. Now, I want to say that before Herceptin and Gleevec came along, we knew that breast cancer patients had amplified HER2 genes, and we knew that CML patients had Philadelphia chromosomes, but we didn't have any medicines to treat them. Okay. The idea here is to get the medicines now, so that once we learn more about how to subset patients, we'll more effectively treat them. So my kind of vision of the future, and I don't know how long this is going to take, that's why it's hard to say. We're going to be able to take tumors out and really analyze pathways that are up and down regulated and customize these therapies. Whether that'll be 1% of the patients, 10% of them, I can't say. But what will result is more effective therapy, fewer side effects, which is really what we want to do. Okay, now for the acknowledgments, now this is the first time I've ever given a science talk where I didn't do one single experiment. Okay, so it's like embarrassing, but these are the people. Well, I'm not a professor, so I can't, Here are the people that really have contributed to this, and of course there's lots of names underneath here. Kayvon Chokot is the founder of IntelliKind, one of the most brilliant chemists I know. He's a great person. Troy Wilson's the CEO. Susan Molyneux is the founder of Proteolix. I should have mentioned Craig Cruz, whose lab the original compound came out of. And she and I basically started this from scratch with Phil Whitcomb, who unfortunately passed away a few years ago. And I should say that company was recently purchased by Onyx, who's developing the drugs. And Oncomed, which Michael Clark and I started about 8 years ago. Tim Hoey is the VP of Oncology. Austin Gurney is the genius VP of Molecular Biology who makes all the monoclonals. John Lewicki's the CSO. Michael Clark is a professor at Stanford who his lab has made the initial cancer stem cell discovery and is continuing along those lines. And my friend Paul Hastings, who's the CEO. Thanks.
Thank you very much, Larry. Those pathways seem very complex. Seems to be like bringing a symphony together. So I think that the Renaissance background is very appropriate. If people would stand up to the mic, I do have one quick question for Larry, and I think it's relevant to looking at the broader context. What we're really talking about here, if everything works, is again taking cancers and, in a sense, converting them into an ongoing chronic condition involving ongoing therapeutic intervention of one sort or another. Not curing, although in the latter presentation, possibly curing. Is that a fair statement, or have I got that wrong?
Well, I mean, the goal is always to increase time to progression. That's our goal. And most of these drugs don't work very well, and the time to progressions are a few months. There are examples like Rituxan where people are on the drug for years, and that's really the goal, is to turn it into a chronic disease like rheumatoid arthritis. And of course, when that happens, then safety becomes more of an issue. So that does play into these ideas of getting a higher level of specificity.
I had a couple of comments, Larry, after the about animal models of tumors, and part of the things that my lab does is we work on glioblastoma, and people have been working with rat xenograft models now for a long time, and but the outcomes in glioblastoma remain. Usually you read the word dismal in most papers on brain cancer. But the thing that really made the difference in our group, we've switched to actually treating people's dogs, pet dogs that get brain cancer, just like people. And now we have dogs that have lasted more than 3 years after diagnosis because we actually went into real tumors in animals that have real spontaneous tumors. And I think that one of the things for anybody who's thinking about working on diseases like cancer, I think the use of pet dogs is a fantastic opportunity that is just not really being exploited. And there's so many breeds of dogs that get a lot of these diseases. I mean, there's a lot of cancers in dogs, right? So I was wondering if you've thought about
Well, I mean, I think obviously there are huge issues with the kinds of animal models we do. And spontaneously occurring tumors are always the way to go. I hope there's no PETA people in here, but when Oncomed does their experiments, the mice run for the hills. The numbers of animals you need to get statistically significant data in these kinds of experiments is quite large. So I think for very, very kind of late stage things, when you're ready to go into the clinic and you want some proof of concept, it might be worthwhile doing spontaneously occurring tumors. There are new animal models which are more spontaneous and less xenografty too, that are being made all the time. But until we understand a lot more about what really starts the tumor and things, how it gets going, it's risky to really depend even on a spontaneously occurring model. Although I will admit it's much better than xenografts.
So, great talk, Larry. I had a question. Apart from cancer, or beyond cancer, when we start talking about pathways and start mapping diseases onto specific pathways, and we're looking for these kind of critical nodes that we can address with some kind of intervention, and then in discussions with people that are making drugs for a living or compounds or antibodies, the word undruggable comes up over and over in certain things. And I wonder if now, in fact, if there is really such a thing as an undruggable target.
So of course there are many undruggable targets.
And what makes them that?
Well, I'll give that lecture at 4. No, I mean, that would be a long talk. Essentially what we like to focus on are things with ATP binding sites, because those have been shown to be useful targets, things like that. Protein interfaces are very hard. There's lots of really tough drug targets. So what we try to focus on are things where we know enough, and there's enough structural data, actually, X-ray crystal data, to allow us to make better predictions. That's where somebody like Kayvon Shokot is such a great scientist, because he's so good at doing things like that. But yes, there are many undruggable targets. But it's getting better all the time. There's new technologies to help there.
Yes, I'd like to ask a non-scientific question, but an issue that came up with Dr. Williams' talk yesterday, and perhaps you can address it, and I apologize if it comes up later in the program, but concerning intellectual property and protection of intellectual property, and the recent case of Myriad Genetics, where the judge ruled against Myriad, which may or may not hold up in future court cases. But could I get your view on intellectual property? And Dr. Williams talked about potential significant hurdles in developing drugs.
So I'm actually strongly against patenting genes and always have been. I think it destroys innovation. It's a bad idea, and I think the Myriad thing is right. What we patent are compositions of matter for drugs. So all of these things I talked about, the antibodies, the proteasome inhibitor, the Intellikine, these are all patented compounds. That's what we patent. I think we don't want to destroy innovation. The object here isn't to make a bunch of money. The object here is to make a bunch of drugs, then make a bunch of money, okay? So I want drugs for certain targets, and if I can't go to a target because somebody has a patent on it, that's just not right. So Genentech tried to do this and it failed too, which I think was the right decision. We don't want to destroy innovation.
You mentioned the class you're teaching at UC Berkeley. I'm interested in finding out more what department that's in,
Yeah, it's
what composition of
It's in the
students are in
business
that
school,
class.
but I don't know anything about business.
So do you only have business students,
No,
or
they're all,
is it open
they're
across
engineers,
campus?
they're engineers and MCB students. There's like 2 MBAs.
Okay.
We can talk about it later. I'll just back.
Could you comment on the change in paradigm? It seems to me that we need a change in paradigm, a sort of paradigm shift in terms of phase 1 trials of these molecular targets, where we're dealing with issues that gets away from the traditional maximum tolerated dose in phase 1 trials, where we're looking at these agents that often don't have an MTD, and really needing to look at targets and how do we assess that, make sure that we saturate the target, but we don't superdose, that we exceed that target.
Yeah, this is an absolutely great topic, which you might think about having next year. The concept of trying to get efficacy in phase 1 is hugely important, both from the standpoint of the patient as well as from the standpoint of the company. And proteolycs, for example, had the luxury of being able to get efficacy in phase 1 because in multiple myeloma, unlike any other tumor, if you see a drop in a surrogate marker, that actually translates to survival. But there's almost no other tumor where that's the case, and I know Larry and everybody else is trying to do this. But the FDA needs to be aware of the fact that the more efficacy we get in phase 1 with these drugs for deadly diseases, other ones, RA, things like that, too much safety problems. But here, for deadly diseases, we need to get more leeway on what we can do in phase 1 and how long and how big and all of those questions. Surrogate markers, these are all really important questions, which, of course, this whole meeting revolves around.
I'm curious about the biology behind using the proteasome inhibitors. So my question is, why would you expect a better therapeutic index with a proteasome inhibitor than with something like a cytotoxic, given that you would think all cells would be depending on proteasome function?
Well, the embarrassing thing about the proteasome inhibitor is nobody knows why there's efficacy. One hypothesis is that myeloma cells are cranking out gigantic amounts of antibody all the time. And if you disrupt, because of that, you need a very efficient mechanism to get rid of unfolded proteins, misfolded proteins. So they're highly dependent on the proteasome. I think that's as good a hypothesis as any, but it hasn't been shown to be the case. And unfortunately, unlike the other pathways, you'll notice I didn't mention a mutation or anything in the proteasome pathway. We really don't understand yet why this is the case, although I think the misfolding hypothesis is easily the best one out there.
So related to that then, it seems to be particularly active in myeloma, but not in other tumor types?
Yes, myeloma is the main one. For example, in solid tumors, it's been dismal, although that may be also because we can't get the dose up because of the side effect problems.
We're going to take one more question.
Well, we have to take Tom's question.
Larry, that was a terrific talk. What I thought you were going to say was that there was a companion diagnostic that was accompanying some of these very targeted therapies. For example, that there was a stratification of patients in the clinical trial, that the Wint pathway was more important or the Notch pathway was more important. And you didn't say that, and so could you just comment on whether you're doing that or not?
Well, it's obviously it has to be done. Those pathways are new and early and complicated, and we don't know enough yet in those pathways, Wnt and Notch, to really know what the right genes are to look at. I think one of the first ones that's going to come to pass here is the Pi3 Kinase Pathway, where you can really dissect all of the components. We know all of the mutations. They're very common in many tumors, and I think that's actually a great one for a diagnostic company to do. Because there are a ton of Pi3 kinase reagents in the clinic right now. So if we can subset, and of course, as I said, IntelliKine is taking patients where we know that those tumors have high likelihood of having the mutations. But we aren't doing molecular diagnostics.
50%.
Yeah, I know.
If you could do the clinical trial
Yes.
just on those 50%,
Right.
that could be really important.
I totally agree, and this is a great opportunity for a group of diagnostic companies to go after these pathways. I think Pi3 kinase, though, is the one I would do first, just because of the complexity of the other ones. Okay, thank you.